everyone, welcome back to our channel Ignited Medicos. This is DEEKSHA, and in this series we'll be learning about amino acids and protein metabolism. And particularly in this video we shall be learning about transdeamination process. We have to know few basics before entering this concept, and they are in this video I'll be using A bar as amino acid and K bar as keto acid. Right. Let's get into the basics first. As we all know, many amino acids combine together to form proteins, and when we break down the proteins, we'll get amino acids. And there are few terms. One is protein turnover. What is protein turnover? Generally, in our body, what happens is protein is not static, it is dynamic. In the sense, protein will be synthesizing and proteins will be breaking constantly. Usually 400 grams of proteins will be making and breaking in the process of metabolism in our body. And what is amino acid pool? It is the free amino acids present in our body at any given time, and that is 100 grams of amino acid pool is seen in our body. And let us look at the amino acid structure first. There is carbon at the center. It has NH2, that is amino group at one end, a carboxyl group, COOH group at other end, and we have hydrogen and we have R. This R can be anything, any group can be attached here, and this structure is called amino acid. And when we remove this NH2 group, when we remove this amino group and insert keto group here, it is called as keto acid. Generally what happens is when we remove amino group from the amino acid, we get keto acid. And in the same way, when we add amino group to keto acid, we'll get the amino acid back. This is what I have written here. When we remove NH2 group from the amino acid, we'll get keto acid, and this is reversible as well. What happens to this NH2? This enters urea cycle. As we all know, ammonia is very toxic, and in the liver, ammonia will be converted into urea and excreted through urine. So let us see two terms which are very important to understand the whole process, which we'll study later. The first one is transamination. When we break down the words, we'll understand the topic or the word easily. Transamination in the sense there is transfer of amino group from one compound to other compound. In this example, we can see there is transfer of amino group from amino acid to keto acid. We'll study this here. And what is deamination? D in the sense we are removing. When you look at the term dehydrogenase, we are removing hydrogen. D in the sense it refers to removal. So as there is amination, what we are doing is we are removing amine group from the compound. It is called deamination. And when we are transferring amine group from one compound to other compound, that is called transamination. So let us look at small example here. As I said, when we remove NH2 group from amino acid, it will be converted into keto acid. In the same way, here we are adding this NH2 amine group into one keto acid, and that will be converted into amino acid. As I said, this is reversible as well. Amino acid will be converted into keto acid, and keto acid will be converted into amino acid. Everything depends on the NH2, whether NH2 is removing or adding. So with this basic, we can jump into the catabolism of amino acids. What is catabolism of amino acids? It is breakdown of amino acids. Catabolism in the sense breakdown. Anabolism in the sense synthesizing. The two terms catabolism and anabolism is together called metabolism. Okay, now let us see how these amino acids are catabolized in our body. And let's see what are the steps first. The first step is transamination, where amino group is transferred from one compound to other compound. The next step is oxidative deamination, which we'll learn further. And then the last step is deamination, where we are removing the amine group from the compound. And these two processes together is called as transdeamination. Now first let us see what is transamination. Before learning what is it, we should know why is it important in our body. The first thing is it helps in the degradation of amino acids, and the second important thing is it helps in synthesis of non-essential amino acids in our body, and the third important point is it provides energy to our body. The next important step is it plays role in gluconeogenesis in our body. And what is transamination? As I said, there is transfer of amine group from one compound to other compound. This definition looks bit complex, so let's see the diagram first. Here, amino group is transferring from one amino acid to a keto acid. Here, this amino acid is converting into a new keto acid, and this keto acid accepts amine group from this amino acid and is converted into new amino acid. Now let's see the definition. It is transfer of amino group from an amino acid to keto acid, converting the original keto acid to a new amino acid and original amino acid to a keto acid. And remember, there is no liberation of ammonia in this process. We are just transferring it and we are not liberating ammonia in this process. And you can easily remember the enzyme used in this process, that is transaminase. As the name suggests here, it is transaminase and it is also called as aminotransferases, as there is transfer of amino groups, right? And the coenzyme used in this process is PLP, or it is called as pyridoxal phosphate. As I said, you can focus on pink written ones and green written ones. Here, amino acid 1 is transferring into keto acid 1 and keto acid 2 is transferring into amino acid 2. You can understand this with example easily. Usually, all amino acids undergo transamination process except lysine, threonine and proline. We can see the example of aspartate and alanine to understand this transamination easily. Aspartate will be converted into oxaloacetate and alanine will be converted into pyruvate. You can remember as SO, and LP, SOLP. You can remember this as mnemonic. And here the enzyme used are AST or SGOT. With the enzyme name, you can easily remember the reactants and products. Usually in every reaction of transamination, alpha ketoglutarate is involved. What it does is, it is converted into glutamate, and it accepts the amino group from amino acid, that is aspartate, and it itself converts into glutamate. And with this process, this aspartate is converted into oxaloacetate. This enzyme is aspartate transaminase. There is transamination of aspartate amino acid. So it is these two are the products, that is glutamate and oxaloacetate. SGOT means serum glutamate oxaloacetate transaminase. These two enzymes are actually same. We can either call AST or SGOT. And in the same way, the second example where alanine is converted into pyruvate, and along with that ketoglutarate, alpha ketoglutarate is converted into glutamate. Here the enzymes are ALT or SGPT, alanine transaminase or serum glutamate pyruvate transaminase. You can remember as LP as I said, and SO. When aspartate comes, oxaloacetate is formed, and when alanine comes, pyruvate is formed. What is the significance of these enzymes? What is the clinical significance? These enzymes are diagnostic enzymes. In the sense, we'll study now. ALT is rich in liver, right? Alanine transaminase enzyme is rich in liver. You can remember this as liver L, and ALT has L. In the same way, AST is rich in heart and then liver. So when there is increased concentration of AST in blood, then that person has high possibility of getting heart attack or liver diseases. And if there is increased concentration of ALT in blood, then the person has high chances of getting liver diseases. Now we have seen the transamination process, and now let's get into oxidative deamination of glutamate. Here in this transamination we got glutamate, right? What happens to this glutamate we'll see now. Here this glutamate is undergoing the process oxidative deamination, where we'll be removing amine group from the glutamate, and in the same process oxidation is occurring. Oxidation is removal of hydrogen. Here the word dehydrogenase refers to removal of hydrogen. Wherever the suffix ase comes, it refers to enzyme. Here glutamate is involved, so glutamate dehydrogenase is the enzyme. This is how you can easily remember the enzyme. What is happening in this process is we are removing amine group from this glutamate to convert it into alpha ketoglutarate, right? Here, as I said, when we remove amine group from any amino acid, we'll get keto acid. So in the same way, when we remove amine group from this glutamate, we'll get alpha ketoglutarate. This is kind of mind map for the things which we have learned right now. The amino acids are converted into alpha keto acids by the process of transamination. In the same process, the alpha ketoglutarate will be converted into glutamate. This glutamate will now be transported into liver, and this process is occurring in all tissues. This glutamate wants to this glutamate enters the liver to undergo this process called oxidative deamination. We'll get ammonium from this process. This ammonium will enter the urea cycle, and then we'll get urea from there. Now let us see what is the significance of this enzyme, that is glutamate dehydrogenase, in our body. As we can see in this process, it plays a significant role in catabolism of amino acids, and it also plays important role in synthesis of non-essential amino acids, and it also plays important role in producing energy in our body. In this way, amino acids are catabolized in our body in tissues, and we get glutamate. This is then transported into liver. In the liver, we'll be deaminating, that is, we'll be removing ammonia from this glutamate. And this is how we come to end of our video. And you can get all these notes in our Telegram channel. Telegram handles will be linked in the description box. You can check them out as well. Please do like, share and subscribe to our channel. Stay positive, work hard, make it happen. Thank you for your patience, guys.